today we will begin with a new unit. If there is any revision that we will do based on whatever we completed last unit 4, unit 3, we will do it just before your exams. Semester exams are there. We can do a revision for all the techniques and stuff that we discussed in the previous 2 units. Now we are going to move up to unit number 2, which is general principles of biochemical investigations and centrifugation. Can you hear me, and is the presentation visible?
Yes, ma'am.
Okay. So when we were talking, we have already seen the very difficult part, that is protein purification, or we have seen that what are the ways of separating or isolating or purifying the analyte. Analyte is anything of your interest, a biomolecule of your interest, because we are going to look at the biochemical aspect of it. So we're talking about biochemistry. Biochemistry, it is a series of chemical processes, and it happens in living organisms. A cell is called as a factory. A cell, you can call it as a factory of reactions, a factory where a lot of chemical reactions happen, and you get different end products, and those end products can be useful. So they can be biomolecules. Okay, so these biomolecules are basically, why are these biomolecules produced by the cell? You can leave an answer in the chat box, or you can put on your mic and tell me, because anyway it's afternoon. Everybody must be feeling a little tired, so let us make it a little more interactive rather than just me speaking and you're listening. Why do you think is a biomolecule necessary? Why does a cell produce a biomolecule?
And for metabolic activities.
Yes, very good, for metabolic activities. Also, you must remember that there are so many pathways. Have you heard about the word? Have you have heard the word pathway? Do you know what is a pathway? You must have heard about glycolysis pathway, or you must have heard about the Krebs cycle. At least these two are very commonly heard, read, studied, isn't it? During your 12th standard, or maybe during the first year of BTech. So there are different pathways, and these different pathways are known to produce some different biomolecules or metabolites, and the end product of one pathway can be the starting product of another pathway. Okay, this is a very important sentence that I'm telling you all, is the end product of one pathway can be an important material, and that acts as a starting material for the other pathway. So these are running in parallel, these are running simultaneously in the cell. It's not that if pathway A is running, some other pathway has to stop till pathway A finishes. Yes, if pathway A is producing a metabolite or a biomolecule which can only start the next pathway, then of course the next pathway has to wait. But at the same time, there are so many other chemical reactions happening in the cell. They are independent of each other. Some of them are dependent on each other. Now, what is the concentration? What is the number? What is the quantity of biomolecules that are produced by the cell? So the cell is doing n number of reactions. So in a minute, the cell is going to produce not many biomolecules. Now, looking at the cell itself, the concentration of the biomolecules, you can't expect that they are going to produce something in kilograms or no tons or something like that. No, they are not going to produce these biomolecules in such large scale. They are going to produce them in minute amounts. And minute amounts are really minute amounts. Like when you analyze them, when you assess the quantity of these biomolecules, they are extremely minute. So basically, we are going to discuss about how we will analyze these biomolecules in the minute cells. So how can they be detected? We have already studied 3 techniques. Can you all tell me which were those techniques that we discussed in the last few classes? What were the techniques that we discussed, like that we use to detect the metabolite or your biomolecule or your analyte? What are the techniques?
Chromatography.
Correct. One was chromatography. Then,
spectrophotometry, and electrophoresis.
correct. Spectrophotometry and electrophoresis. Correct. So actually now you will find this topic very easy because we have already discussed the difficult part before. So we should have gone unit 1, 2, 3 and 4 like that, but we came unit 4, unit 3 and now we are at unit 2, but this is going to be easy for you now. So we have already discussed these techniques, chromatography, spectrophotometry, electrophoresis. So what was the main aim, or what was the aim of doing all this? The main aim of doing all of this was to isolate the analyte of your interest, to isolate the analyte or to isolate the biomolecule that you are interested in as a scientist, as a R&D person. You are interested in extracting that analyte from a particular uh, reaction. Okay, because that analyte may be industrially important, that analyte can be a very important component in some fertilizer, or that can be one very important metabolite in a drug. So you need to now isolate it. Why you have to isolate? Because when we are doing a reaction, when we are talking about these chemical reactions that happen in a cell, do you think it is only the biomolecule of your interest which is produced? Do you think that only that molecule or only that analyte will be produced and nothing else is going to be produced?
No, ma'am.
No. So because there are so many other reactions going on, so many other pathways working simultaneously in that particular cell. So not only your biomolecule, but there will be other cellular components also. There can be some components which are broken cell walls, which are absolutely rubbish. They can be like a dead cell fragment or anything like that. As well as if the cell has lysed, it will just release everything out into the medium. Or if the cell is still alive, it is just secreting the biomolecule, it is throwing it out, but along with the biomolecule there can be other smaller fractions which you do not want, other cellular components also which you do not want, you are not interested in that. So obviously if I have a mixture which contains biomolecule, so I have this particular mixture here, I have a container here, and I have, so these are my biomolecules of interest. Somehow I was able to get them. Now, along with these biomolecules of my interest, I have these little cellular components also. Okay. Now, the green one is of my interest, but along with the green one, I have the blue one, I have the red one also. So, is the, what will you say, is this clear, is this a pure mixture sample, or is this a mixture, is this a sample mixture of a lot of other things? Of course it is a mixture. So this mixture indicates that my interest, the interest on the biomolecule of my interest right now is along with some contaminants. So the red particles, the red biomolecules or the blue components, they become contaminants. Okay, they are the contaminants. So how will I be able to obtain the biomolecule? These ones, what should I do in order to obtain the biomolecules? I must separate them. I must separate the biomolecule from the contaminant. Okay, so this is what we do in biochemical analysis. We plan a strategy, we design a method to only retain our interest, the biomolecule of our interest, or the analyte of our interest, and the rest of the stuff which can be contaminant. Even if it is useful, it can be useful for some other thing, but right now for us it is not useful. Right now it is causing contamination in our pure sample. So that's why we have to remove that. So the other material has to be removed, and the biomolecule of your interest needs to be isolated, needs to be purified. So now what classifies as a biomolecule? We are talking about biomolecules now for last so many classes. What classifies, what is biomolecule? Any molecule that is produced by a living organism. We know that in the living organisms there are so many chemical reactions that are happening at the same time. So as an outcome of those chemical reactions, there are some products that are formed. Those products are biomolecules because it is in a living cell. including large macromolecules. They can be proteins, polysaccharide, lipids, nucleic acids, and they can also be small molecules also, such as primary metabolites, and they can be natural products also. So these things are known as biomolecules, and they are important. They can serve as a raw material for for another cycle, or simply they can be extracted and they can be used um, for any industrial purpose. Okay, what are the types of biomolecules that we will now look at, and see, based on these types there can be different strategies that can be developed for the separation of these biomolecules. So the types of biomolecules are lipids, proteins, polysaccharides, glycolipids, sterols, then glycerolipids. Okay, vitamins are there, hormones are there, neurotransmitters are there, metabolites, monomers, oligomers, polymers, in short, carbohydrates are there. So these are the types of biomolecules which are produced by the living organism. Can we move ahead without knowing, analyzing, detecting the biomolecule? Can you go ahead, like you have been given a task now that you have to produce a particular, you have to isolate a particular biomolecule named A. So you do a reaction, you set a reaction in the lab, you do that experiment. Now can you, at every step that you are going to do, will you be able to move ahead or plan a strategy, whether I should do a chromatography, whether I should do an electrophoresis, whether I should do affinity chromatography, or what type of chromatography, spectroscopy? So to plan all of that, is it not important to know what biomolecule you have? Yes or no? At every step of your experiment, is it very important to know that what biomolecule is being produced? Is it of your interest or not?
Yes, ma'am.
Yes. What will happen if you just blindly work? Suppose you are working on production of protein. Suppose you have done all the experimental setup to produce protein. But if you do not check whether that protein is being produced or not, and at the end of everything you find out that it is not a protein, it is some carbohydrate, then is your experiment successful?
No, ma'am.
No, it is not. So obviously at every step, so to move from step A to step B, to move from step B to step C, you must know that at every step what is being produced, whether the biomolecule is being produced, whether the biomolecule concentration is increasing or decreasing, whether your biomolecule that is being produced is active or inactive. Okay, because it might happen that in the first initial stages it is active, later on it can become inactive. So you have to keep on checking all of that, and then I would say you can also conclude that your experiment is successful because you have been able to get this particular biomolecule in a high concentration, in optimum conditions, and in an active form. If I am doing a certain experiment, if I'm running a certain batch, if I get a good amount of biomolecules, suppose it's like too good, but if that biomolecule is not active at all, is that important? Then is that experiment successful? If I get the biomolecule in a very good quantity, nobody has reported that, that much I have been getting due to my experimental setups. But if that biomolecule that I get is not in the active form, is it a successful experiment then?
No, ma'am.
It is not. So what happens is you have to keep on checking whether the analyte, the biomolecule, the metabolite, whatever it is that you have been producing, the cell has been producing, or you have been meaning to produce in that particular medium, is active, is produced in whatever concentration that you look at. Okay, so you need to have that strategy designed. Now, biomolecule analysis is very, very important. So you run multiple number of experiments, but if you don't have an analysis technique in place, it is very difficult to conclude anything. So these techniques, biochemical analysis techniques, basically, these are very important. And these techniques refer to set of methods. It is not only one method that you will be taking up to. It is a combination of many, many methods. So these techniques of analysis will include assays, and you will be using a spectrophotometer. Probably you will be doing, after you do a spectrophotometry, then you might analyze it. You might take the analyte and do a chromatography, you might do electrophoresis. Okay, so assays are very, very important. We are going to see the definition of assay also in the next slide. So these techniques, the biochemical analysis techniques, they refer to a set of methods. It's a cluster of methods. It is just not one method, it is a cluster of methods like assays and procedures that enable the scientists to analyze the substances found in living organisms. Now, these organisms are producing these substances as an outcome of ongoing chemical reactions. Now, these chemical reactions are catalyzed by the enzymes in the cells. So, they are the biochemical reactions that happen. And as an outcome of these reactions, there are substances that are produced, and these substances need to be analyzed by the scientist. Now, how the scientist is going to analyze, there is no one certain method, but there are cluster or there is a group of methods that you're going to take up to analyze this particular substance. There is a need. Of course, you will have to come up with a strategy. The scientist will have to design a strategy to detect the biomolecule. He or she will also have to first isolate it in the pure form. So once you have the biomolecule isolation, we have studied column chromatography, electrophoresis. We have seen this, we have seen the technique. So you can use either of the technique depending upon your biomolecule. Whatever is the biomolecule, that will determine that what you are going to use. Are you using column chromatography? Are you using electrophoresis? What are you doing here? You are basically separating, you are basically isolating that particular biomolecule in its purest form. Now, why do you need the purest form? Of course, because when you have a pure form, you can do a lot of other studies also on the pure form. Okay, sometimes you will observe that a biomolecule is the most stable and the most active in a pure form. Or sometimes you'll also see a very contradictory situation where the biomolecule loses its activity in the pure form. It is in the pure form, but it is not active, it is not functional. So such a thing is not going to help you. So you need a strategy to detect the biomolecule first, whether it is produced in a particular reaction or not, and then isolate in the pure form, which is contamination free, and then do the further studies. Further, you can use that particular biomolecule to study the structure of it, the function of it. If it is a drug metabolite, then you can study interactions with different salts, metals using that metabolite. So you must have a pure form. Now, whether it is pure or whether it is active or inactive, that the assays will tell you. Okay, we are going to see what is an assay. So there are certain analysis techniques that we have already, one or two, three of them we have already studied. The other three that are mentioned is radioimmunoassay, ELISA, and centrifugation. Centrifugation is going to be covered in this particular unit. So we have studied how spectrophotometry helps us in the analysis. So what do you get? What does spectrophotometry tell you? So if I give you an analyte and if I tell you to do spectrophotometry, so what can you tell me from the data that you get from spectrophotometry?
Ma'am, concentration.
Very good. Concentration. Then any other? Functional groups?
Yes, ma'am.
Yes. Okay, you can also comment upon the functional group also based on the behavior of that particular analyte at a particular wavelength. Okay, what do you do in chromatography and electrophoresis? Yes, what do you do in the, what data do you get of your biomolecule or the analyte when you do chromatography?
Ma'am, density.
Chromatography, okay,
Ma'am,
density,
size.
size, correct. Size is the proper word. Then think about all the chromatographies, right? Paper chromatography, dekha, we have seen column chromatography, size exclusion. In column chromatography, we have seen size exclusion, affinity chromatography, ion exchange chromatography. So you can comment a lot about the size, the charge. Ion exchange chromatography is based on charge. Do you remember? So you can comment about the charge on the protein, right? And electrophoresis that you do, again, it's a ratio, charge to mass ratio. Okay, so you can get a lot of idea about in this first three techniques that that is there on the slide: spectrophotometry, chromatography, and electrophoresis. They will help you to step by step proceed towards the final conclusion about what the biomolecule is. So by spectrophotometry, you can actually detect whether the analyte is actually there or not, the biomolecule is there or not. If it is there, then what is the concentration of it, and what are the functional groups based on whatever wavelength you're going to use. Okay, chromatography, you can comment upon the size. Then you can also talk about the charge, mass ratio. Here also you talk about the mass, charge. Okay, so if it is DNA, then you know that that is negatively charged, so it is going to go towards the positively charged electrode, positive electrode. And you can use SDS gel electrophoresis page for proteins. So you can extract the protein in the native conformation. When you do a native gel, you do not distort the 3D structure of the protein. When you do an SDS, you linearize the protein. The SDS is imparting negative charge on the linearized protein. So you can actually learn a lot of characteristics of the biomolecule. So analysis techniques are very, very important. And as I told you, it will depend upon the biomolecule only. So the biomolecule, if I have lipid, then what technique will I use? If I have a lipid molecule as my molecule of interest, okay, so can I do agarose gel electrophoresis for that? Now if I have lipid, can I do agarose gel electrophoresis? Yes,
No, ma'am.
no. Okay, Aditi also says no. Yes, you cannot do agarose gel electrophoresis depending upon the metabolite. But now if your biomolecule is a nucleic acid, what will you do then? If it is a nucleic acid, if it's a DNA or an RNA, then of course you'll go for this. So your biomolecule is going to tell you what to do and what not to do, which analysis technique that you should take. Okay. Now, before you perform any biochemical analysis, before you proceed, there are certain prerequisites. Matlab something, these things are required before you step on to analysis. The first is the first step is strategy to detect the biomolecule. You must have a plan as in how you will detect the biomolecule. Now you know certain things like starch. Okay, you must have heard about this test where you add iodine to starch. Then what happens when you add iodine to starch? You'll have done this probably somewhere in 9th standard, 10th standard, Thai. Black, blue, black. Yes, it turns blue, black, correct? So now if I have to determine, or if I have to detect a biomolecule, it's a polymer, it's a starch. What is the simplest test I can do to detect starch? I will just add iodine to the test tube. So if I have a test tube now and I did some reaction happened, and then as a result of that, now it has some biomolecule. I suspect I am looking for actually to produce starch. Okay, so if I want to confirm that there is presence of starch in here, the easiest of all is I add a few drops of iodine to it, and then if it turns bluish black, then I know that there is starch. So this is the strategy that I came up with to determine starch. If there is a reducing sugar like glucose, then there will be a different test for that. The Felling's reaction is there, or there are so many other reducing sugar tests. So glucose is a reducing sugar. So there are so many other tests that you can do to determine glucose. If there is protein in that, then there is okay, yeah, if there is protein, so what would you do if there is some protein as a biomolecule? The first thing that you can do is chromatography. At what is the wavelength for proteins and nucleic acids? What is the specific wavelength for proteins and nucleic acids?
200 to 400, and for DNA 250.
Yes, yes. So 200 to 400, 260, 280 nanometers. So one is for protein, one is for DNA, one is for nucleic acid. So yes, this is the basic strategy that you build up when you have to detect the biomolecule. It does not have to be perfect, but to start from somewhere, you need to come up with a strategy. Okay, then I will do a UV spectrophotometry, or I will do a visible spectrophotometry, or I can do an electrophoresis, or I can simply do a starch test, that is that iodine test. If it is a reducing sugar, then I can do some reducing sugar tests. If it is glucose, then I can do a certain assay to find out whether there is glucose or not. So this is the strategy designing point. Okay, once I detect that there is a biomolecule, okay, I know now, yes or no, yes, it is there. The test tube turned blue black, so starch is there. Okay, now what I do is, now along with starch, I know it's a chemical reaction that is happening in a living cell, and I also know that that is not the only biomolecule that will be produced. There will be other contaminants also along with starch. So my first job is now to isolate. My second job, sorry, the first job I did was to detect the biomolecule. So it is there. Now it means that I must isolate it. I must extract the biomolecule. I must clear off all the contamination, and then I will be able to study on the purest form of my biomolecule. Okay, so isolation. Now isolation is when, if at all you are doing a protein column, or you did say you did UV spectroscopy, and you found out that yes, protein is there, you got a good reading on the spectrophotometer, and then you have detected that this biomolecule is protein. Let us now consider that this biomolecule is protein. So you have to isolate. How will you isolate this protein in the pure form? Which is the technique? We have studied that technique. Which is the technique that you are going to use to isolate the protein in the pure form? What will you do to isolate the protein in the pure form?
Chromatography.
Chromatography. So yeah, you be confident. It's correct. It is chromatography that you are going to do. So chromatography, you have size exclusion chromatography, you have ion exchange chromatography, affinity chromatography. So all of that you can choose either of the process, either of the column chromatography technique depending upon your biomolecule, and you can isolate the protein in the pure form. Okay, so if you're doing a gel exclusion chromatography, then what will happen? The large molecules will flow faster. Okay, the small molecules will get entrapped into the beads, right? So that is one strategy. The other strategy is ion exchange, where the separation happens on basis of the charge that is exchanged. Affinity is highly specific, so you have a certain ligand that is attached to the matrix, and then based on that you can also isolate the protein. Now after the isolation of the protein in the purest form, you have to characterize the biomolecule. So characterization is knowing further more details about the biomolecule. So first, isolate the pure form. Okay, you isolated the protein. Now let us say that this protein is say amylase. Okay, so if this is an amylase, you need to further characterize it. So if you have to characterize it, then you must know what are the, is there a functional group, or I would rather use the word catalytic site. Okay, amino acids that are involved in the catalytic site. We know that the proteins are made up of amino acids. So obviously this amylase is also going to have amino acids. There is going to be a catalytic site. Do you know what is a catalytic site? You know that no, you know that every enzyme has a substrate.
Yes, ma'am.
Yes, every enzyme has a specific substrate. Okay, so what is the substrate for amylase? A known substrate for amylase? Starch. Okay, have you forgotten about salivary amylase, which is there in your mouth, which is so important in the digestive system that it breaks down starch? So the enzyme acts on the substrate, it breaks down the substrate into smaller fragments. So here amylase acts on starch, it breaks it down into smaller monomers, because starch is a polymer. So it will monomer, dimer, okay, it will break it into small pieces. Amylase now has many other substrates also. So starch is there, amylose is there, another substrate. Amylopectin is also one of the substrates. And then maltose is also there. Okay, so it has many substrates. And with every substrate, the reaction, of course, it is going to hydrolyze only. What is hydrolyze? It is breaking down the substrate molecule. Okay, it will break down the substrate molecule into smaller pieces. So it is it falls in the class. There are some 6 classes of enzymes. So amylase falls in the hydrolases. Okay, hydrolases is cleaving or breaking, the action of cleaving or breaking. So the amylase enzyme breaks the substrate into smaller fragments. Now when I characterize amylase, what will I do? I'm going to look at the reaction. So I will take the amylase which is in the purest form, and I will set different reactions with different substrates. So I may take like 4 different test tubes. In first test tube, starch. In the second, it will be amylose. Third, it will be amylopectin, maltose. And I'm going to add the same quantity of starch to all of them. I'm going to look at the reaction. I'm going to study the reaction. I'm going to then do spectrophotometry and analyze how much product is formed, because you know it's like a chemical reaction. When enzyme reacts with a substrate, it forms a product. So what product is formed? What is the quantity of the product? So with what substrate does amylase work the best? I can get that information. So this is, I am doing is characterization of the amylase, characterization of a good substrate, or I'm characterizing my amylase for the best substrate. So I can study the best substrate for amylase. That is also characterization study. The other characterization study, okay, so the catalytic site is a site where the substrate comes and binds to an enzyme. So like if this is the enzyme, okay, with a cleft here like this. So this is the substrate. So this is the site where the substrate is going to bind. So it means that this is the catalytic site of that particular enzyme. So I can study, when I characterize it, when I characterize the enzyme, I will study that what are the amino acids involved in the catalytic site. Okay, so that is another characterization study that you have to do. Then there are other further characterization is what is the optimum pH of that particular enzyme. Do you know that all the biomolecules, see the biomolecules are produced in living cell. Okay, we just saw that. Now the living cell is functioning at a certain pH. What is the pH and what is the temperature at which the cells in your body, in anybody's body, is functioning at right now? What is the temperature? Suppose we are talking about the human body. What is the temperature of the human body? 37. Okay. What is the pH? Near to neutrality. So it means that all the cellular reactions that are happening right now in the human body are at a temperature between 35 to 37 and at a pH which is near to neutral. Okay. Now, if we think about a bacteria, if we are working on a bacteria, of course, the bacteria will also have an optimum pH at which the bacteria can produce enzymes, and whatever enzymes and protein it is producing, the protein is stable at that particular temperature. Okay, so when there is a change in the pH and when there is a change in the temperature, the biomolecule happens to become inactive. So the biomolecule is very sensitive to pH. It is very sensitive to temperature also. Okay, very sensitive to ionic strength also. If you are using a particular buffer in the lab, if you're doing a column chromatography, if you're doing any sort of study on the pure form of that biomolecule, it will be sensitive to pH, it will be sensitive to temperature and ionic strength. So change in pH. Now, salivary amylase. Do you know what is the pH of salivary amylase? Okay, salivary amylase is in the mouth. I'm just taking this example because you have already studied this in the school. Even if you have not studied much about enzymes in this particular course, you will study, I think next year maybe, but you must be knowing what is salivary amylase. Where is salivary amylase produced?
In the parotid glands.
Correct, very correct. So what can be the pH of it? Neutral.
Neutral.
Yeah. Then as you go down in the digestive system, there is stomach. So stomach may, what is the enzyme there?
Pepsin.
Pepsin. So pepsin, is it neutral, acidic or alkaline?
Acidic.
Acidic, correct. What is produced in the stomach? Hydrochloric acid. Okay. So initially, this is pepsinogen. Initially, it is pepsinogen. In presence of hydrochloric acid, it becomes pepsin. Pepsinogen is inactive and pepsin is active. Okay. So then, now if you swap the places, if you make pepsin, if you put pepsin in the mouth, and if you put salivary amylase in the stomach, now salivary amylase will have to face HCl and pepsin will be at neutral pH. Do you think that these enzymes, when you swap the places, will be active?
No, ma'am.
No. So it means that pH is a very, very important factor here. Again, going into the small intestine, there is another enzyme that is trypsin. Okay, so that is from the bile secretion that comes, and the inactive form of it is trypsinogen, but in alkaline pH it becomes trypsin. Okay, so this is the active form. So again you will see that the salivary amylase, the pH is neutral, pepsin, the pH is acidic, and trypsin, the pH is alkaline. So you see that there are three different enzymes here, and three of them have different, they are functional at different pH, which means that it tells you that proteins are very, very sensitive to the change in pH. If the pH changes, they become inactive. If the pH increases or decreases, they become inactive, or their efficiency reduces. What is the meaning of efficiency? See, every enzyme is functional when it acts with a substrate and it produces a product. So this is a functional enzyme. Okay, so what happens is enzyme plus substrate will give you an enzyme substrate complex, and then this will give you the product. This is a product. Okay, and after the product is formed, then the enzyme is released and can go further reacting. So this is known as an active enzyme. But if because of the change in the pH, now what will happen because of the change in the pH? Now there are certain amino acids which are present in the catalytic site. So when the pH changes, it means that the amino acid in the catalytic site will have a different pH. These amino acids will probably change, and the whole shape of the catalytic site also changes. Because of the shape of the catalytic site changes, then the enzyme will not be locked with the substrate. The substrate does not find a proper fit with the enzyme, and hence the efficiency of enzyme to form an enzyme substrate complex and then further form a product reduces. Okay, so pH is one very, very important factor. The temperature, again, so our body is functional at 37. So all the reactions, cellular reactions happening at 37, we feel very comfortable when our temperature is 37. In high fever, when the temperature increases, loss of appetite happens, you start feeling weak. Okay, you don't feel like eating. Even if you are eating, the body metabolism is not so great. Why? Because the temperature has increased. It means that the cells are now subjected to high temperature in the body. So it means that the cellular reactions that are happening at a higher temperature are happening very slowly. So the rate of transformation of whatever you are eating into a particular product is so low that you are feeling low in energy. Okay, so temperature is again a very, very important factor. If you increase, so if an enzyme like all these amylases and this pepsin and trypsin, whichever are there as a part of the digestive system, all of them are functional, say, at the body temperature between 35 to 37. If the body temperature increases beyond that, then these enzymes will also work, but they will not work with their fullest capacity. So that also affects. So temperature is one of the major causes for enzyme inactivation or loss in the efficiency. Ionic strength, so the buffer that you are using now, this buffer and all that, when you do experiments in the laboratory, when you are doing a column chromatography especially, you're going to use buffer there. Okay, so if you remember the question that was asked in the paper about the steps, arrange them in a proper order. I don't know how many of you all have opted for the right option, but there was something like sample loading. The sequence was like this, the correct sequence: sample loading, binding, no, first was equilibration of column, then degassing of the matrix, then sample loading, then binding, then elution. Do you remember this question which was there in the question paper?
Yes, ma'am.
Yeah, I don't know how many, what you all have chosen, but this is the right sequence. Like, you first equilibrate the matrix with the buffer. When you make when you equilibrate the matrix with the buffer, now that buffer is the buffer which has the pH required for your enzyme or required for the protein, required for the biomolecule to have its optimal activity. So if it is salivary amylase, and if I want to purify that, then I will equilibrate the matrix at pH 7, because salivary amylase is stable at pH 7. I will not equilibrate the matrix with pH 5 or pH 9. I will use it at pH 7 because the biomolecule is stable at pH 7. Then you degas, you remove the gas, you fill, you pack the column. After packing the column, you load the sample. After loading the sample, you're going to bind it. So you allow the protein sample to travel through the column, and as it is traveling, it will interact with the matrix and it will bind, or the larger molecules will run through the void spaces, the smaller will enter into the matrix depending upon what type of column you have packed. After the binding has happened, then you will elute the column. Now elution is a process where you are using salt. You may change the pH. You may change the ionic strength here. Okay, so in the step of elution, you do salting in, salting out. We are going to see this also in this particular unit. We are going to discuss more about salting in and salting out. We are going to, so what happens once you bind the protein to the column, once you bind it, it should not bind irreversibly, then there is no use. If it is if the protein is binding to the matrix permanently forever, then how will you separate the protein? How will you remove the protein from the column? So at the step of elution, you will have to change the conditions. So what will you do? When you change the conditions, automatically the protein will come out. Okay, so what will you do? You might add more salt to the buffer. So because of the salt, the interaction between the matrix and the protein may weaken and the protein will elute out. Or you may change the pH, because now suppose at pH 7 the binding is maximum, very strong binding. Okay, so while elution, what I do, I use a pH 7 buffer plus I add salt. Salt means, say suppose I decided to add NaCl to it, and then I try eluting the uh, protein from the column, but still it is not happening. Why? Because at pH 7 the bonding is very strong, but if I alter, if I change the pH 7 to pH 5, then the protein might leave the matrix and come out of the column. So pH change is very also plays a very important role. And then change in ionic strength. So while binding the protein, if I had used 10 millimolar of buffer for binding of protein, then if I now change the ionic strength, so instead of 10 millimolar, if I elute the protein with 1 molar, which means 1000 millimolar. So instead of 10, I have used 1000. I have increased the ionic strength by 100 times, 100 fold. If I change the ionic strength of the buffer and then I elute the protein, it might elute. So sometimes altering these conditions will help to weaken the interaction between the matrix and the protein. But in such cases, what am I also doing is I am posing a threat to the protein also, because when the pH is changed and the ionic strength is changed, the conformation of the protein, the shape of the protein might also change. If the shape of the protein changes, the catalytic site also changes. If the catalytic site changes, then there is not enough enzyme substrate interaction, and the protein will not form enough products after the enzyme substrate interaction. So ionic strength plays a very important role. If I increase the ionic strength, then the conformation might change, leading to change in the catalytic site, leading to change in the interaction between the enzyme and substrate, and it is difficult to get a product there. Okay, so characterization of a biomolecule is not only one thing. There are so many several things. You can find different substrates. You can select the best substrate based on how much product is that substrate forming. You can also study what is the pH. So sometimes it is like the protein is neutral, but it can still tolerate a high pH. So till pH 9, it might give you a product. The efficiency can be low. So all that data you can use, and you can provide that data to the industry where you're handling the biomolecule, and then they can use those studies to design or formulate whatever product they are making. Suppose you're making, if you're working on a protein amylase, and that amylase needs to be added to a detergent. You know that detergents also have bioenzymes in them. Detergents have lipases, amylases, proteases. Why? Because the food stuff that you eat, that we all eat, sometimes the stains on the clothes that are there is mostly of oil, some food particles, and all the food is made up of, say, complex polysaccharides, lipids. So they will be easily cleared by using enzymes. So you can use you can use these enzymes in detergents. So now when you're using these enzymes in detergents, and detergent is a highly alkaline pH there also. So you can see that the protein that you are using should be stable at a high pH. Okay, it must be stable at a high pH, plus it must not denature in the presence of detergents, because we saw SDS is denaturant. SDS is going to linearize your protein. Similarly, when you add the bioenzymes to a detergent, that will also have the same effect on your biomolecule. It will deactivate, it will denature your biomolecule. So all these studies, characterization studies, that how stable it is in the presence of a surfactant, how stable it is to change in pH, how stable it is to change in temperature, to ionic strength, all these studies come under characterization of the biomolecule. Okay, there is biochemical characterization and there is biophysical characterization. When you talk about characterization, there is biochemical, biophysical. In the biophysical characterization, you can do CD, circular dichroism, to comment upon what is the conformation of your protein. So after you subject it to different pHs, you can take your protein and you can add it to different pH buffers. And then after keeping them for, say, some time, you do a CD, and you can then have a data that at pH 9, the conformation was distorted. Okay. At pH 7, the conformation was intact. It was tertiary structure. It was intact. But as the pH increased, the conformation disturbed and it was distorted. So these are some studies that this is also a part of biochemical analysis based upon the biomolecule that we're using. If the biomolecule is lipid, everything changes. Then I do not talk about enzyme substrate complex, or I do not talk about this and all that. That is altogether a different thing, because now the biomolecule has changed. If your biomolecule is a hormone, then again, everything changes based on the biomolecule. I cannot use the same strategy that I use for protein when it is when the biomolecule is a hormone or a metabolite or a natural product. things change. The strategy also changes when the biomolecule changes. Now, once I detect the biomolecule, I isolate it in the pure form, then I characterize the biomolecule, then I go for analysis of its function. It's very important to determine the function, and along with it, the concentration also is important. So these are the two things which can be achieved by doing an assay. I told you that we will look at the term assay. So here is the definition. What is an assay? It is a biochemical test that characterizes a molecule, whether quantitative or semi-quantitative. So when you do an assay, you actually find out the function of that particular biomolecule, as well as you also find out the concentration of the biomolecule. Okay. Now, for example, again, we will take a bacteria. Now that bacteria, you add it to a medium. Okay. So first, when you have, this is the container that you have, and this is your medium inside. Now in this, you will have initially, like this is the just the beginning of a reaction. You have added some bacterial cells. Okay. After 24 hours, so the same number of cells is going to increase. Okay. Now you tell me, this is the 0 hour and this is the 24 hour. So if this bacteria is producing the biomolecule, where is the concentration? At which hour will be the concentration more? Will the concentration of the biomolecule be more at 0 hour, or will it be more at 24 hour? Yes. 24 hour. Yes, correct, Mitali. So after 24 hour, obviously the concentration of the biomolecule in this particular container will be more. Why? Simply because the number of bacteria have increased. So number of bacteria increases, so number of the product also increases. The product is your biomolecule. So as time progresses, the concentration will increase. But I told you about the stationary phase, where what will happen, all the nutrient will get exhausted and there is no further nutrient available for the bacteria. So if the nutrient is not available, then the product will also not be formed. So there will be a time, suppose that time is 72 hours. So at 72 hours, there will be no change in the concentration of the biomolecule. Even if the number of bacteria is a little slightly more than that, but then you will see no change in the concentration. Then you must expect to see a graph. So if you now plot a graph on the formation of the, or on the concentration of the product, you will see something like this. Okay, so if you want to plot a graph, it will be concentration on the x-axis and time on the y-axis. Oh, sorry, x-axis is time and concentration is y. So you will see initially what will happen as time passes. So say suppose this is 0 hour and this is, sorry, this is not working. Okay, if this is, sorry, time, and if this is concentration, so you have 0 hour. This is the time where you have initiated the reaction when you added the bacteria to the culture. Then let us say 2, 4, 6, 8, 10 and 12. Okay, so you will see nothing is produced here. At 2 hours also maybe you will see nothing. At 4 hours you are going to slightly see some reading here. Okay, when you do, you just have to take the a little amount, aspirate a little amount of the broth and then do an assay. Okay, when you do the assay, you will find the concentration is this much. Then after 6 hours, maybe it has gone up. After 8 hours, it has gone even upper, like this. 12 like this. So what will happen if you try to plot? You will see that it will go up, 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 and then there will come a point where it becomes stationary. It is not going to increase. It is not going to decrease. It is just going to remain stationary. But if you leave it for a longer time, the biomolecule starts degrading also. So the concentration might start going down because of the degradation. But now the bacteria is not growing anymore. So this can be somewhere near between 60 to 72 hours, because all the nutrient from the broth is exhausted now. There is nothing food, no food left. So no bacteria is going to grow, no bacteria is going to form any product. So you will see a stationary phase, you will see a plateau. Okay, this is the kind of graph you see. So this an assay can definitely tell you. So what do you do when you are running an experiment in the lab? The most important thing is to find out the concentration of the biomolecule. You will have to intermittently take samples out. Okay, you have to decide that at every hour is not possible. Why? Because every hour, if you're going to take out the sample, you are going to cause more and more contamination also. Secondly, if in a flask you're only taking 50 ml of broth, and if every hour you're going to remove 1 ml, soon you will be left with nothing in the flask itself. Okay, so every hour is not possible. Maybe after every 6 hours that you can decide. So after every 6 hours, you come to the lab. In the sterile conditions, you put on the laminar airflow, you take out the flask, and then you take out a small sample from the flask, you centrifuge it, allow the cells to settle down, and then take the supernatant and do an assay. If you do an assay, you will be able to get some color. You read that color in the UV spectrophotometer, okay, and then you find out what is the concentration. So when you're talking about concentration, you're talking about quantity. So a biochemical assay is quantitative or it is semi-quantitative. Means it is not going to give you a full idea of how much is the concentration actually. It is not very accurate, but it will still give you an estimate. Okay, it is very, very important to determine this quantity and the presence of the biomolecule at every step. So that is why you should know that when is the time when the bacteria is producing the biomolecule in the highest concentration. So when is the time actually, at what time? Is it 24 hours? Is it 48 hours? When the concentration of biomolecule is the maximum in the broth, that time you should know, and probably that is the time where you can stop the reaction and then further start processing the sample. Okay, you need not run the whole thing for 72 hours or 80 hours, because after a particular time, when the exhaustion happens, no change is going to happen. So there is no need to run the experiment. So you must keep on doing this assay to find out a time point where the concentration is maximum, and once you detect that, you can stop the experiment and you can harvest it. Okay, so this is all about biochemical analysis as an introduction. We are going to also see centrifugation. We are going to look in more detail aspects of other analysis techniques also. So when we are doing centrifugation, what is the logic behind doing centrifugation? How can you achieve separation? Or when you're doing electrophoresis, in short, again, chromatography, in short, you see that what are you trying to achieve when you choose for those kind of analytical techniques. Okay, so I will